So thank you so much. It's a great pleasure to be here. I hope all the presentations are bringing new ideas, and I'll be presenting what we've been doing in our research in this area of brain data analysis. And well, I will try to be technical, but not very technical, try to be in the middle so the public watching the video now and in the future can follow, right? So, well, just as a brief presentation, my background is in bioengineering. My research area is more focused on that connection between engineering and biomedicine. And basically, I work more in the software side than the hardware side. So I use, in our lab here, we focus to use more sensors already developed or some under development, but our main target on research is on the software side. This is a nice picture from one of our campus here. USJ is a Catholic institution in Macau. It's I believe Macau is a region that is China, but it's not China. It's a special administrative region, and we are in a handover process from Portugal to China that will take 50 years, and we are now in the 22nd anniversary. So here at USJ, we have our laboratory was created. You have our website here, and then you can visit to see what kind of research we've been doing. But our lab was created in a mix because, as you can see here in the figure, we are allocated in the Faculty of Business and Law. Why? Because we started here 4 years, almost 5 years ago, with a very clear target of developing one laboratory of neuromarketing. And we have some research. Some of the results I'm going to show today are very connected to neuromarketing. But then with the maturity, right, and the development of the research group, then we started to go through different areas, and we created the lab, officially the Laboratory of Applied Neuroscience, and we are connecting our research with many other faculties here, like education, psychology, architecture as well. So we have a mix of devices and software that we've been developing, and like eye tracking, skin response, EEG, of course, right? Heart rate as well, and facial expression. So several different devices. I'm going to explain a bit better in the future slide about what we have. But if anyone is watching the video or here would like to know more about the lab, it will be a pleasure to develop joint research and also receive master and PhD students here. So just a very fast background. We started here in 2017, and then we've been increasing the capacity of our systems. The first thing we started was EEG and heart rate, because actually my research areas, my publications were very focused on the heart in the beginning, directly ECG processing and heart rate variability. And then we started creating new approaches and using new technologies, new devices. And then today we have comprehensive, not complete. We still need a lot of things to add to the lab, of course. But I believe we have a a group of solutions that allow us to create several possibilities of research. So we have corporations established in different countries. I have PhD students in Portugal under my supervision, in Brazil as well. We have a cooperation with the Boston University and some other universities in China. And these are some of those I highlight here. This SIAT, which is one key institution here in Shenzhen, very close to Macau. I also work there as a visiting academic. And we have the Universidade Católica, which is a Catholic university in Portugal, which is like a mother university of USJ. And then we have other corporations, very active actually, with these other universities and institutes. So I already highlighted some of the areas, but just as an example, we've been doing some things connected to the BA programs, like because here we have undergraduate, master, and PhD programs in business. So we have been studying not only neuromarketing, but also something more broader, right, that we call neuroeconomics. We have been working with stress, right? A few months ago, we had one conference in stress, but I presented another research cooperation with Brazil. But here in the lab, we have one research going on related to stress and decision-making that is quite interesting, the results. Papers are coming soon, I hope. So we've been working directly with consumer behavior. Then we have some students from psychology, also cognitive development, metacognition. We've been trying to work with metacognition in the lab. So if anyone here wants to do something related to that, we are very keen to open this. We are going to have the first PhD student in the area now. And then communication, with Faculty of Communication and Media. And also there is an interesting approach related to culture and heritage. You know, this is to answer one need, because Macau is a very touristic region. And as you might know here, the key tourism is the gaming, right? So we have the casinos, very famous, 7 times bigger than Las Vegas, right? So it's but the government wants to diversify strongly, right? Create new things. So the cultural tourism, this connection between Portugal and China, West and East, is something that is attracting a lot. The government is pushing a lot, and obviously the heritage is one key aspect. And then we are developing this research in this area. So going to our specific topic, I'll control my time here, otherwise I will be too long. 1 second. Yes. So this is the title of my proposal, right, for this talk. And I separate here in 3 different parts. That is application of multiple supervised and unsupervised or non-supervised. And it's the first thing, right? Then machine learning, artificial intelligence, techniques, and direct and indirect brain monitoring system. So I'm going through these 3 colors as the guideline for the remaining of my presentation. So we actually going to start with the end, right? Let's give a brief discussion about brain monitoring systems. And I put here 3 possibilities of BCIs, of brain-computer interfaces. Of course, we have famous image systems like fMRI, which is a reference, right, for many types of analysis, or if you want to go more to the diagnostic side or the medical side, the fMRI, of course, is a key reference. But we have in our scope here, right, I'm bringing here to a more a narrowed scope. I'm bringing here the idea of analyzing or monitoring the electrical activity of the brain externally with the EEG. We have here one FNIR system, which is monitoring the brain activity, but with optical sensors, right? This is something that we are trying a lot to bring to our lab, one FNIR's solution. Basically, actually, we want multimodal. I'm going to talk about this here, I think. And FNIRS generates image, right? It estimates the brain activity according to the level of oxygen in the blood, in the brain. So that's where the region that you have more blood, right? And then basically is with the EEG, we can have a very good time resolution, but we lose in spatial resolution. And here is the opposite. We have very good spatial resolution, and we don't have a good time resolution. So both together is a very promising approach. And obviously, we also have electrodes, right? Electrical monitoring the electroactivity of the brain, but with implanted electrodes, right? So this is for more advanced research, basically medical analysis or medical applications. So from the EEG, I believe everybody attending this kind of session is a bit, at least knows what the EEG means, and this very complex signal in the time domain usually is transformed into something more easily readable in the frequency domain, right? So we have here the power spectrum of the EEG, and we have the wave bands. Basically, most of our research are focused on alpha and beta intervals that we're targeting a lot about stress and cognition. And then when I start talking about indirect brain monitoring, we can automatically remember about the ANS, right? The autonomic nervous systems, which we have the two subsystems called the sympathetic and parasympathetic nervous systems, which create both of these systems. They are all the time interacting in our body, trying to balance our reactions and our well, basically all the many physiological responses of our body. So here in the middle we have one very easy to detect measure, which is the heart rate, and the heart rate is influenced by the sympathetic system. Basically, when it accelerates, right? So the sympathetics wants to accelerate the heart rate. The parasympathetics, on the other hand, wants to slow the heart rate. So this balance creates what we call the heart rate variability, which is, if you see here on the top, we calculate the distance between the R peaks of a QRS and of the heartbeat. Of course, here I'm using the ECG, but it's also possible to use the PPG, and so you have the peak of the PPG and you calculate the distance between the two peaks. So this gives us lots of information about the subject condition and generates many different metrics. we were just running some tests on a new database that we have. Is a ECG database of COVID patients, and we are analyzing the heart rate variability. So just the first analysis, we could generate for each patient something around 85 metrics, right? Only using the heart rate variability. If we go, then this is important. This number of metrics is relevant when we start using computational systems, like such as AI systems, right? And then for the brain, we're still in this direct brain monitoring. One requirement, I would say requirement, was one decision that we made in our research group here was that our target was always to try to find the simplest solution so we could have very realistic experiments and setups, right? So for example, we have here one fMRI, and of course is a fantastic tool, very reliable, everything is okay, there's nothing against it. The problem is only the setup, right? So it's when I want to analyze stress, for example, the position, the machine, only the environment is already a strong stressor here. And also the situations that I can generate here for the subject are, I would say, hard to control, to create a baseline to analyze stress, right? So we started analyzing different types of devices. Then we have here some screenshots from the Emotiv, which is a very famous device. I believe everybody here knows Emotiv, right? And then we have 2 devices in our lab. And then we started going to non-medical. Emotiv is also non-medical, right? But we also work with medical EEG, of course. In our research in Brazil, recently, what we've been doing was we were executing one EEG, quantitative EEG in the beginning, and one quantitative EEG in the end. And then we were using these simplified devices for neurofeedback sessions, relaxation sessions, and cognitive analysis. And then this simplifies a lot the setup so we can have simple things. So we have this BrainLink device, which is from one company that we work here, is sponsor of our lab called Microtellect. And there are several possibilities that we've been developing. For example, this is a spider that we connect with the brain device, and then we can, according to your level of focus, the spider can walk. If you're more focused, it will walk faster. And then this is one example that we created a race between 2 spiders, and it was quite interesting to see. And then that's where comes the idea of the metacognition. It's amazing to see how each subject develop different approaches to achieve the level of concentration that is necessary to make the spider walk. We have different devices that we can control. For example, this is a lamp that changes the color. And then there's this, I just put this here because this is the opposite, right? In this case, we monitor the level of relaxation, right? So the lamp changes from purple to white and among several colors in the middle. And the more relaxed you are, the closer to white the light goes. So today we have been doing these multiple technologies together, right? Why? Because sometimes for one setup that we develop, the EEG becomes hard to analyze. That's why we need maybe what I like to say is that they are supporting metrics. For example, when we use here in the core biometrics, if we use skin conductance, right? So the GSR, we cannot classify anything, right? But we can detect emotional arousal, right? So peaks, just those peaks they can be treated as starting points for the analysis, right? And here in the core biometrics, we also have the heart rate, where we calculate the heart rate variability. Here in the facial coding, we use algorithms that automatically classify emotions according to the facial expressions. And the eye tracking, of course, in some experiments, eye tracking is extremely useful so you can analyze what is attracting the attention of the participant, right? And then we have a module of self-report where we can survey the participants and see what they respond, right? For example, in the stress experiments, we've been always running the self-perceived stress test, right? Which is an international measurement. Okay, yes. So this is the big picture of what we can think about direct and indirect brain monitoring. And from the machine learning and AI thing, there is always a big mix of concepts, right? And then I just bring here, I just selected some diagrams from the internet, so we can give our contribution to the area. And people with less contact with the technical side can understand a bit better, right? So artificial intelligence is a very broad concept that brings many different approaches, such as knowledge modeling, symbolic reasoning, model logics, and we have here in the middle, we have the machine learning, right? Inside the machine learning area, we have different types of approaches, right? I'll bring specific slides here about that. And then we hear a lot about data science, right? So there is one intersection here, right? We have the data science side that is not artificial intelligence. When we are doing, because we see a lot of people saying about regression techniques as part of machine learning, right? So that's wrong. Statistical analysis as part of machine learning. That's not correct. Machine learning is when you use something that make the computer learn something, right? Do some kind of decision, and from this decision, learn something or change how it will work in the next step. So this one is interesting because inside the machine learning, we have the idea of the brain-inspired systems, where we have the famous neural networks, right? And deep learning is one technique among different types of neural networks, right? I just brought here one very fast slide about this. We have the artificial neural network that try to follow the biological model of the neuron. The neuron receives lots of connections the same way as the neuron receives, transmit the information, and the neuron only transmit information if it is active, right? So here in the artificial neuron, we have one activation function that it will only transmit the information if the neuron is active. So we just put all of them together in what we call the network, creates lots of connections because the magic of the neural system is the endless number of connections, right? Billions and billions and billions of connections. So this creates the power of processing, right? And then in the middle of this, we have the concept of deep learning, but I'm not going to talk a lot about this here. Just highlighting that deep learning networks started to be used very successful with image recognition. That's why you see lots of techniques and even time series being transformed into images and then analyzed by deep learning solutions. So for supervised, and the third part of this, we have the concept of supervised and unsupervised, because this is key for what we've been doing in our research. So just a very simple example you can understand. When you're talking about supervised learning, you're going to train your network, your model, right, according to reference, right? This is what we call the input data. We are going to tell the system that these input data here are apples. And then when I submit something new to the model, right, the model will be able to recognize that this sample here belongs to this classification. So it will tell us that it's an apple. In in unsupervised learning, actually, we have a bunch of data, and our model will try among these mixed data to create what we call clusters, right? It's a clustering process. So this is very important for us because many of our analysis, we don't have previous annotations, right? We have our input data, we have several different variables, and we just try to identify clusters from this input data. So this is one key contribution for us. If our clusters are reliable, according to other metrics that we can analyze, then our system becomes reliable. And then we have a bunch of algorithms here. I'm not going through this a lot. I still want to present my two researchers. I'll be fast, I promise. So the number one is one AI system based on EEG. EEG basically in the beginning, but then we expanded to other metrics afterwards. The idea here was we were analyzing user reactions when watching different types of videos, right? So the experiment was designed, well, our sample was 30 participants, 50-50%, different nationalities. We didn't set any criteria for nationality. It was one experiment inside the university, so we had exchange and local students. And then the methodology was, we wanted to test the user engagement, attention, through different types of videos. But then we used two videos related to Macau tourism because of the largest project related to the Macau tourism here with the government. And then we have two, one with 3 minutes and one with only 1 minute, that we call the opening video and the closing video, that we wanted to see the user reactions to these. And then in between, we had a random experiment here. This is random, so the sequence is not fixed. We had one context-less video, like someone talking about something that there is no connection with here, like agriculture in one country. We had two short, funny videos. We had one contextualized video talking about the region here, And then we had one inspirational ad to see what happened. Then in the beginning, we only monitored the EEG, and then afterwards we started a second phase of the experiment, doing more data connection. And then in our software, we started to analyze some statistical parameters. If you check here, this plot on the top is the attention level of the participant. And then we separate the experiment in three we separate the participant in three the experiment in three parts, and then we have three parts here: the beginning, the middle, and the closing, right? And we started running some visual and statistical analysis, and we started also calculating some other metrics. In the end, we had 23 metrics from here, and we did the same thing for the relaxation level, right? And we started afterwards running some clustering analysis because we didn't have any kind of pre-classification of our participants, right? We didn't have anything that we wanted to compare with. We just wanted to see if we could create clusters of participants according to their brain activity. And we could separate some of our metrics here. This is one general figure, but it's quite similar to what we got. We use some dimensionality reduction techniques like PCA, so we could reduce, in this case, the number of metrics for the most relevant, and then we could find three classes that we could not. Of course, you can see here they're not completely separable, right? But they indicate different levels of participants, right? This is a very promising research. We've been, well, this experiment was the basis for our software development. Everything is still under development and validation, but I believe soon we are going to have the first official publication about that. We already have been doing some conferences and science popularization about that, and hopefully soon we have the most concrete results. Now we have one master student doing a second experiment, not related to Macau Turing video anymore, but very focused on marketing strategies. Well, for the second example I wanted to bring here, just to close my presentation, is, well, this one was not collected by us. This is a database that is available. It's about consumer reactions using EEG. So we have, this database was collected using the emotive with 14 electrodes, right? 14 sensors. And usually the participants were exposed to the stimuli and they needed to select like or dislike, right? Very simple experiment, but lots of exposure, lots of brands and products and objects in the experiment. So for this one, I'll focus more on our methodologies because I have one PhD student that is creating different approaches for this, specifically this data set, right? So I want to go through this so you can understand how important it is for the machine learning technique that we do something before to extract good features for the model, right? So that's why we have here the description of the EEG data set, right? It's 25 participants. 4 seconds, the person exposed for 4 seconds for each product, and then it needs to select like or dislike. And the idea is to use the EEG data to see if we can predict, right? If in our brain we are already saying something that that person liked or disliked that product, right? So we have a phase here of pre-processing, right? We were analyzing different bunches of data. So we have the full data. We were analyzing first 60 seconds and then divided in blocks of 60 seconds. And we were applying some filters, but we are doing much more now. This is a very simple filter. And then in this model here, what we've been doing was we were extracting features, right? So we were analyzing 67 features per channel into the time domain and the frequency domain, and calculating some nonlinear metrics like entropies. Basically, it was only sample entropy, approximate entropy, those famous time series entropies, but now we are planning to use different ones. And then for the machine learning approach, we were doing, first we were applying the PCA, right? And we were calculating or extracting the feature importance, right? Based on random forest. Random forest is a tree, is a decision tree technique, right? Algorithm. And we were calculating the statistical testing and doing the basic analysis. After we started doing a second approach, so we wanted to use deep learning, right? In this case, to use deep learning, the pre-processing is the same, but then we transform the data using CWT, continuous wavelet transform, and generate images for each data. And we submitted the image to different types of deep learning architectures, right? Very promising results on this one. And finally, it's like a next step of the first one. Actually, it's not really a new model. You can see everything the same. We have all the features. We do like feature importance. We perform our statistical testing, and then we go to the classification layer, in which we are testing completely different approaches like SVM, KNN, XGBoost, random forest, MLP, naive Bayes. So different approaches, some decision tree approaches, some different, I would say, requirements and different approaches, I think is the key word. And we are comparing the results and the performance of each of these. So now at the moment, we are also doing some filtering approach with ICA, independent component analysis, and the results are going to be published soon as well. So what is our next step to conclude my talk here? As I mentioned, we are planning a lot to go to the multimodal monitoring. Actually, we need to expand our brain monitoring capacity in our lab. So we are probably buying this year one multimodal EEG FNIRS device, right? So we want to have both and analyze. Of course, we don't need 20 electrodes, but we need at least 8, so we can have a good mapping of our brain activity. We are also working with some research groups on the genetic mapping that will be together with our physiological monitoring. And obviously, many of our research, we have several different clinical analyses that we use as markers for the analysis. For example, this one that we are analyzing the COVID patients, we have enormous number of clinical tests and inflammatory markers and blood samples. So this is powerful to support our AI classifier. Yes, and then I finish here. Thank you for your time, and I'm available if we have any question.